Hi class, in this recording we're going to focus on bone cells and bone tissues. So let's start off with our precursor cell. This is a form of stem cell called an osteogenic cell. Osteo meaning bone, genic meaning to create. And as we look at these osteogenic cells, these are multipotent stem cells that are found in the endosteum, that inner membrane of the cell that lines the medullary or marrow cavity and or surrounds the individual spikes or fibers, technically known as trabeculae, within spongy bone tissue. And we also have osteogenic cells on the outer membrane of the cell, the periosteum. All of these osteogenic cells are going to arise from embryonic mesenchyme, one of the germ layers that we talked about. And as we look at these osteogenic cells, they're going to continuously multiply and give rise to all the other cell types of our bone. So here we have our favorite osteogenic cell, just a little osteogenic cell. And that osteogenic cell will first morph into an osteoblast. Osteo meaning bone and blast meaning build. I want you to think B for build. Blasts are a kind of cell that build. This particular blast builds bone tissue, osteoblast, bone building cells. Now as we look at this osteoblast, they're going to have a single layer of cells underneath the endosteum and the periosteum. And they're going to continually synthesize that organic matter, the soft organic matter of the matrix, in particular the collagen, lots of collagen fibers and protein fibers of the matrix. And after synthesizing that soft organic matter of the matrix, they're then going to have mineral deposition. The mineral deposition is going to be the addition of calcium and phosphate salts or crystalline compounds, also some carbonates mixed in as well. Now as we look at these osteoblasts, they are going to stimulate osteogenic cells to multiply rapidly. In particular, the stress being placed on these osteoblasts and osteocytes will stimulate osteogenic cells to multiply rapidly and increase the number of osteoblasts. So when we are physically stressing our bones out, we make more bone building cells, and by making more bone building cells, we make our bones bigger and reinforce them so that those bones are better able to withstand the stresses that we're putting onto our bones. What does this mean practically for you? If you want to increase your bone density, you need to stress out your skeleton by applying force to your skeleton. This could be with something as simple as going for a bike ride, gardening, exercise, just something that moves your body. It doesn't need to be brutally miserable in order to reinforce your bones. Any physical activity will stress your bones and reinforce them. Now, at the end of the life cycle, eventually we end up with the osteocytes. The osteoblast turns into the osteocyte, site meaning cell. So an osteocyte is just a generic bone cell. What ends up happening is that osteoblast will continue to excrete extracellular matrix, will excrete the inorganic and the organic components of the bone, and eventually they bury themselves. They become trapped in that extracellular matrix that they were excreting. And once they're completely trapped, they morph and become the osteocyte. As an osteocyte, there's a little tiny cavity that they're going to sit into. That tiny cavity is referred to as a lacunae. And there are multiple lacunae and multiple tissue types. And notably, we have lacunae within cartilage and within dense bone tissue. And then there's little tiny channels that come away from the lacunae. The little channel itself is called a canaliculi. I had some former students almost a decade ago now that instead of saying canaliculi, they would always say, "Can I lick your eye?" And they had a little song and dance routine that went with that. It left a strong impression on me. I thought it was hilarious. It's worth emphasizing that these canaliculi are the channels, the hollow spaces, that extensions of the osteocyte sit within. And those extensions of the osteocytes are going to reach out to a neighboring osteocyte so that they can contact each other. And a lot of times what ends up happening, and I'm going to shift to red here with my pen, is if I apply physical stress to the bone, that physical stress is going to cause cytoplasm to flow in and out of the extensions of the osteocyte. And we can have cytoplasm flowing in to the cell body, or we could have cytoplasm then rushing out of the cell body. And it's that influx and efflux of cytoplasm, or excuse me, cytosol, into the extensions of the osteocyte that acts as a strain sensor. And what ends up happening is that when we physically stress our bones, we activate the strain sensors within the osteocytes, and those strain sensors are then going to initiate bone remodeling. And it's kind of like remodeling your house, where you change the shape and structure of your house. With bone remodeling, you change the shape and structure of your bones just a little bit. You don't have to have enormous changes. Normally, what ends up happening is there's going to be growth of the bone in response to the physical stresses being exposed to. So that growth of the bone could cause there to be a bony prominence growing on the outside, or if it's just overall stress, we could just increase the overall density of the bone. Great coffee. Love coffee. Now there's one other cell type for the bone that we need to talk about, and that's the osteoclast. Again, osteo meaning bone. And when I think of clast, the clast are the destroying cells, the bone dissolving cells. So I like to think, let me clear the screen here for a second here. We have that C for clast. I like to think for C for cleaning it up. Osteoclast clean up the bones and make them go away. An osteoclast is a clast is a cell type that destroys a tissue. What tissue is being destroyed by this particular class? Bone tissue is being destroyed or dissolved by it. So an osteoclast is a bone dissolving cell found on the surface of the cell. Osteoclasts notably come from multiple stem cells fusing together. Those individual stem cells have individual nuclei, and all of those nuclei are then going to be fused or going to merge together, and an osteoclast is one of the few multinucleate cells within our bodies. And within our osteoclast, we have a ruffled border. This ruffled border resembles microvilli in terms of the structure and the fact that it's going to dramatically increase the surface area. And that ruffled border will always face the bone tissue itself. And as that osteoclast has its ruffled border facing the bone itself, it's going to excrete hydrochloric acid. That hydrochloric acid will then dissolve the bone matrix and then create a pit that the osteoclast can sit in. This pit is referred to as a reabsorption bay on the surface of the bone. And as that osteoclast reabsorbs the calcium and phosphorus that was within the bone, it will then release that calcium and phosphorus into the bloodstream so that we can maintain blood calcium and blood phosphorus homeostasis. We can maintain that relatively constant concentration within our bodies. Notably, and I think this is kind of cool, I want to point this out here, the osteoclast is ginormous relative to the osteocyte, which is pretty dang tiny in comparison. So those were our cell types of the bone. Let's talk about the extracellular matrix of the bone in more detail. As we look at our extracellular matrix, the extracellular matrix of bone or osseous tissue, if we were to dry it out, it's about 1/3 of the overall. It's about 1/3 organic and 2/3 inorganic. So it's mostly calcium and phosphorus salts. The organic matter within the bone matrix is going to be made by the osteoblast. Collagen is the big one right here. I can't emphasize that one enough. Lots and lots of collagen. But we also have carbohydrate protein complexes. These glycoproteins are going to be broken up into glycosaminoglycans, proteoglycans, and glycoproteins. And as we look at these categories of molecules, we'll talk about them in more detail later on in the course. The inorganic matter of the bone is the part that doesn't have a lot of calcium. Excuse me, not calcium. Brain hiccup right there. I meant to say carbon. In particular, carbon within amino acids or within carbohydrates. So as we look at this, hydroxyapatite, calcium hydroxyapatite is the overwhelming majority of the inorganic matrix of our bones. And this is where we have lots of calcium and lots of phosphates in that particular mineral. We also have some calcium carbonate, within our bones, and that's a minority at about 10%. And you're probably thinking to yourself, hey, wait a second, Mr. Durst, carbonate has a carbon. It's a CO3 with a -2 charge. Yes, but carbonate, carbon monoxide, and carbon dioxide, even though they have carbon in them, are categorized as inorganic molecules. They're the exception to the rule. So they're still going to be inorganic based on the definition and the noted exceptions to that definition. And then there's also the others, the 5% others. This is where we store lots of other minerals in our bodies. And this is also, from a physiological perspective, where we're going to store heavy metals. If we suffer from heavy metal poisoning, we are going to sequester those heavy metals within our bones. So if you have excessive gold, lead, cadmium, palladium, common heavy metals that people can get poisoned by, well, maybe not gold, but the other ones are more common. I wish I was suffering from too much gold in my life. But as I look at this, it's worth noting that these heavy metals that are sequestered in our bones are removed from circulation. This is one of the ways our bodies handles heavy metal poisoning. The problem happens, and I know this isn't in your notes, but this is, I think, fairly significant clinically. When a woman is pregnant, and that woman is releasing calcium from her bones so that her growing child in her womb, her fetus in her womb, can have mineralization of her bones. Those heavy metals, so I'm going to use red here to represent little lead atoms. Those little tiny lead atoms that have been sequestered in the extracellular matrix are released by this giant osteoclast that is dissolving the extracellular matrix. So what happens a lot of times is when a woman suffers from heavy metal poisoning as a child, she sequesters those heavy metals within her extracellular matrix as she grows larger and expands her skeleton. But when she becomes pregnant, she re-releases those heavy metals into her bloodstream, and some women suffer from heavy metal poisoning because pregnancy is causing those heavy metals that have been hidden in their skeleton to be released back into their bloodstream. Here we go. Let's talk about aspects of the matrix. When we think of the mineral portion of our matrix, that mineral portion makes the matrix really stiff. The protein portion, that organic component, gives it flexibility. I like to think of rebar and concrete. The protein is kind of like the rebar. The mineral portion is like the concrete. Here we can see a bone that's been soaked in vinegar overnight. And by soaking that bone in vinegar overnight, we've removed most of the mineral component, or a lot of the mineral component, and now the bone is super flexible. Here is a bone that was baked in an oven at 300 degrees Fahrenheit for 6 hours. So a nice slow bake, and we oxidized the proteins in it to remove the protein component but leave the mineral component behind. And you can see this bone is shattered very easily. Now as we look at these mineral and protein components, there are diseases associated with these. So rickets, for example, is when somebody doesn't have enough minerals within their bones. Rickets is known as having flexible bones. And those soft, deformed bones, particularly the long bones of the lower legs, think of the tibia and the fibula, and to a lesser degree the femur. Those long bones of the legs are going to have a lot of deformity or flexion to them. When we look at the organic component, some people suffer from a disease known as osteogenesis imperfecta. And that's a really kind of a generic name. Osteogenesis means creation of bone, genesis meaning creation, and imperfecta means imperfect. So if we look at the Latin roots here, it's imperfect creation of bone. That's what the name literally means. More practically speaking, osteogenesis imperfecta means brittle bone disease. And these individuals have a defect in their ability to deposit collagen into the extracellular matrix of their bone. So their bones are, in particular, super duper brittle. They have lots of fractures. As we look at compact bone tissue, and I see that I'm going at 14 minutes here, so I really need to speed this up. Our compact bone tissue has lots of osteons. And here we can see an osteon as a series of concentric circles. Here is a photomicrograph of an osteon, and that within that osteon, in the very center of the osteon, we have a dark circle called the central canal. Just worth emphasizing here, compact bone tissue is also known as dense bone tissue. That's another synonym for compact bone tissue. Let's go back to the osteon. As we're looking at the osteon, there's concentric rings within the osteon, around the central canal or Haversian canal. Those central rings are referred to as, concentric rings are referred to as lamella. And then connecting one central canal to another central canal, we have perforating channels. So here in red, I'm highlighting some perforating channels, or sometimes referred to as transverse channels. And then in green, I'm highlighting a central canal right now. We also have spongy bone tissue. Spongy bone tissue was also known as trabecular bone because the little bone fibers within it are referred to as trabeculae, and a really old school name is spongy bone tissue, trabeculae, and I'm drawing a blank right now. You could typically only find this name if you look in textbooks that were made in the 1960s or earlier. It'll come back to me later. So as we look at the spongy bone tissue, for the sake of disclosure, I'm a spongy bone kind of guy. I think that's a very descriptive name. It's my preferred name. And as we're looking at the spongy bone tissue on this photomicrograph, the bright pink, the magenta portions are the trabeculae. And then the speckled red parts, that's the red bone marrow in between the trabeculae. So the spaces in between are filled with red bone marrow. As we look at spongy bone, it has very few osteons. There are absolutely no central canals. And instead, all those osteocytes are going to be very close to the bone marrow. And there's trade-offs here. Spongy bone tissue is not as strong as dense bone tissue, but the perk of spongy bone tissue is that it provides most of the strength of dense bone tissue, but it has a heck of a lot less weight. And as we look at the trabeculae within spongy bone tissue, the trabeculae tend to develop along the bone's lines of stress. So as you look at the trabeculae in a bone during lab or in a photo of the trabeculae, those trabeculae within the spongy bone tissue form along the stress lines of the bone. That's all we have with this recording. It's a little bit longer. Sorry about that. What can I say? I started sharing anecdotes and rambling on. Hopefully you enjoyed those extra side stories. If you have any questions or comments, please feel free to post them to the class discussion board or to shoot me an email. And as always, happy studies.